came the same month. And as you know, it was also a problem dating carbonates. How do we date carbonates? Shallow water carbonates. What a pain. And one day, Bob comes back from a meeting, I don't know where he was. And he said to me, well, you know, I was snoozing away during the talk, and all of a sudden he heard bacteria and magnetism in carbonates. And of course, he listened to Joe's talk. And from then on, he was convinced that we should now get a magnetometer and start to date the cores and the shallow water carbonates. And that's how it started. So then Dawn, for his PhD, started to measure all the magnetic stratigraphy on the cores, and under inclino, the core's the top portion was basically dated with magnetostratigraphy. That was really the only way we could date it, and still till today it is. And so I'm very happy to introduce Joe Kirschvink, who helped us date the shallow portion of this famous course. Please.
Gosh, everyone, I'm really honored to be here, and it's a pleasure to give a tribute of some sort to Bob Ginsburg. I'd like to do it by reporting a bizarre, maybe, origin of a particular type of carbonate mud in Palau. And it's kind of humorous, but interesting, and it has an underlying theme of Earth's magnetic biosphere. And so I'd like to say that Bob is probably going to be popping up and watching this every now and then. But it's good and it's fun. So I'll give you a brief outline, Caltech style, of what I want to do. First, I want to mention there is a Ginsburg-Kirschvink connection, and it's a note on the Lowenstein scale, actually, because at the University of Chicago, Ginsburg actually had several faculty mentors, one of whom was Heinz Lowenstein, paleoecologist. For those of you in the petroleum industry, you probably know that Heinz Lowenstein, was the guy who recognized the facial paleoecological associations in Silurian reefs of the Chicago area. He was the first guy to really be able to triangulate from industry drill cores the location and position of ancient reef complexes that were oil-bearing. Heinz is the one who really figured out the paleoecological associations of that. Anyway, he was one of the people that really got Ginsburg interested in reefs and things back at the University of Chicago. And then I'll talk about one of the discoveries in biomineralization that Lowenstein started with. Of course, Lowenstein and Ginsburg are advocates of the biological origin of many of the aragonite crystals in pelagic organic carbonate oozes and muds. It's not always stood the test of time and has been controversial, but that got Heinz interested in biomineralization. And that's actually one of my fundamental interests, passions, I would say, particularly the mineral magnetite that we'll talk about. And that really ties around into a study we did recently that gives us the origin of some of the micrites, the muds in the interior lagoons of Palau. And it's a humorous story, and I'll tell it, and it's funny. There's some Japanese who won't like it very much, but you'll see why. And then that leads into a broader story that I'll conclude the last 10 minutes or so of my talk. Earth's magnetic biosphere. What is this mineral magnetite doing? Why is it there? How old is it? And what are its ecological and biological implications? And at the very end, I will show you something about human magnetic reception. I'll spoil my punchline by telling you now that we actually have a major manuscript in review arguing that humans have the same intrinsic neurological ability to detect the geomagnetic field, the same way that honeybees, homing pigeons, whales, bats, and other animals do. So that's where I'm going, and we'll see if I can get there in a brief 30 minutes. Okay, so first, the Ginsburg-Lowenstein-Kirschvink scale. Heinz Lowenstein, again, was a Jewish refugee from Nazi Germany. He managed to get out before the war, just barely. Wound up at the University of Chicago, well, actually in Chicago, where he had completed his PhD thesis at Munich, but the Nazis blocked him from getting his PhD because he was Jewish. But to their credit, the University of Chicago faculty saw the thesis, saw his letters of recommendation that he smuggled out with them. They made him a graduate student. They gave him a fellowship in the middle of, he was destitute, managed to get him his degree, and he wound up working with Harold Urey's team during the war, and among other things. So anyway, but while he was on the faculty there, Bob Ginsburg actually worked with him. I mean, they were a committee of people. And so here's the kind of the Lowenstein scale. There's Mike Nesson, who did his thesis work on chitons. Dick Squires, a paleontologist, came out. Steve Weiner, who joined the structural biology department at the Weizmann, worked on biomineralization and still one of the main leaders in the field of carbonate biomineralization, the molecular basis of how animals actually make carbonate. And then there's me. I did actually a graduate degree with Hans. He was my master's advisor at Caltech. But then I went to Princeton and worked with Al Fisher and Bob Hargraves and Jason Morgan and others. So anyway, that's the scale. So Heinz is approximately first student, or at least one of the early ones in Chicago. I was kind of the last one. And the reason, well, how did I get to know Bob? Well, there's a funny thing. Bob's sister, Mildred, was the spouse of Murph Goldberger. Murph Goldberger was a physicist, was Caltech's president from 1978 to 1987. And so he would come out to visit his sister often. And I joined the faculty at Caltech in 1981. And of course, Heinz said, we got to meet this guy. And that's how I ended up with Ginsburg. And it's how Bob said, oh, Joe, we've got to do this magnetics of these carbonates. And at the time, shallow water carbonates were one of the worst materials for paleomagnetists to study. It was just a pull your hair out mess. But actually by developing new magnetometers, and as Don McNeil will mention, we actually managed to do it. So that was fun. Also, we learned a lot from Bob in those visits. I remember fondly having dinner with Bob, my wife Atsuko and I, and we'd go into the restaurant and Bob would say, Joe, Atsuko, the head waiter wants to put you over at that cruddy little table. Don't let him do it, insist on that table. I had no idea that these head waiters would do that. And it was Bob that actually guided us through it. And anyway, he's a fun guy. Also, another funny story that apparently he and his wife, when they moved to Miami, bought a house, and then every 2 years they would sell it and buy another one and kind of visit all the suburbs of Miami until his wife passed away, in which case he stayed there for the rest of his duration. Anyway, it explains actions of my wife, too. Anyway, so that's the Ginsburg-Caltech connection, and it's fun. So I'll launch into the story of the magnetite story, and this is Heinz Lowenstein standing in the intertidal notch in Palau, at least one of them. A picture I took of him in 1988. And his discovery of magnetite biomineralization is actually classic. When he was working with the US Navy, Caltech faculty during World War II were given ranks in the Navy. Full professors were given the equivalent rank of admirals. And that held on until after the war. And when Heinz moved to Caltech in 1952, that was still active. And so he discovered he could go throughout the South Pacific flying on the military air transport system. And everybody was, oh yes, Admiral Lowenstein, okay. They would fly planes, it was wild. But he used it to study the coral reefs, particularly uplifted ones in Palau. And one of the things he noticed were those notches, which according to the German textbooks at the time, physiographic textbooks, they were supposed to be carved by wave action. Except Heinz noticed that the depth of those notches increased the further into the interior of the lagoons that you got. It didn't match being wave action. And he finally looked and realized that the actual rock in those zones was striated with subparallel grooves. And as he was watching it, the story goes, he actually saw one of these animals, the mollusks, the Chitons, polyplacophorine mollusks, go through and leave a fresh pattern of those scrape marks. So he said, now wait a minute. Again, the major German physiological books said that the teeth of mollusks were supposed to be made of the protein chitin, similar to your fingernails, and yet no way could that be strong enough or hard enough to actually remove the surface layer of algae that these animals were feeding on in the cracks. So that led him to actually do the mineralogy. They took it back to Caltech and x-rayed it and eventually discovered that they were made of the mineral magnetite. And in fact, I have one of Heinz's original I saw that when I was a freshman at Caltech. When I came to Caltech, I didn't know if I was going to be an electrical engineer, a physicist, a geologist, or a biologist. And I spoke to Gene Schumacher about this problem, who was our division chair at the time, and he gave me Heinz Lowenstein as as my mentor. And then Schumacher signed me up as a lab technician student for the USGS paleomagnetics lab he was building in Flagstaff. And I told that to Lowenstein, who was then assigned as my advisor. And Heinz said, oh, you need to see this. Ooh. And I was hooked. So I've been working on magnetite bond mineralization for over 40 years. Anyway, and if you look at those teeth, they're beautiful. They're black, they're dense, there's 600 species, ultrastructure is beautiful. The magnetite crystals are sitting there inside collagen networks, single crystals. Heinz had done a bunch of studies identifying it, again, with X-ray powder. This is a famous paper back in 1962. And of course, when you find one crazy mineral in the teeth of a mollusk, you look for others. You also found goethite in archaeogastropods, again, very hard. And we actually now know that those teeth structures, the ultrastructure, are some of the hardest materials that organisms make. The magnetite and the goethite teeth are extremely strong, and that's exactly what you would expect after 500 million years of evolution for strength. So wild. But what about this funny thing in Palau? So I'll tell you a bit more of the study of the Palau and Palau white and things. Heinz published a wonderful chapter in 1974 in The Sea, Ed Goldberg's monograph series of everything you've ever wanted to know about the ocean, but we were afraid to ask. And Heinz wrote it up in his horrible English, broken German thing. And Goldberg promised that he would edit it and get the English lined up, and he never did. And so it's a good example of Lowensteinian Milwaukee German English, as he would call it. But in that thing, Heinz described these nips, notches at sea level, even to the species that were sitting there eating away and doing it. And so that's kind of where it stayed from about 1975 onward. Just as a regional background, Palau is in kind of 1,000 miles east of the Philippines, in the middle of the Philippine Sea. There's the Yap Trench that comes in, and then the Palau Trench. And Palau are these emergent islands right here. And you can imagine subduction zone, as the plate goes down, it warps up, and the ancient atoll that is settled down comes up and gets exposed and goes down. So it's one of the few places where you can actually go and see a three-dimensional structure of uplifted reefs, including the big island where the volcanic core is actually up and eroded down. So it's kind of a beautiful area. Again, you can see the Palau Trench, the Kyushu, Palau Ridge, the various islands, and then Palau. But that doesn't, and of course Palau is right there. If you look at three-dimensional models, you can see that, oh, well, okay, it's a nice carbonate platform. Sea level goes up and down. But if you actually look at the bathymetry around it, you see it is really stark. The trench edge comes, the 8-kilometer deep trench comes within 40 kilometers of the edge of the reef complex. It's like a 5-to-1 slope going straight down the thing. Things are rolling down. You can dredge blocks of the Palau reef that is slid down, and yet the bottom of that trench is very sharp. There's no infering flat sediments. It goes chunk, chunk. So it has to be extraordinarily dynamically active. Only problem is there's no seismicity, none. Quiet. So anyway, and you can see that. Here's the edge of the reef, and 40 kilometers out, you get it. But as it comes in, the tongue of the seafloor goes up. You get the uplifted reefs, and something weird is happening. So here is an aerial photograph. This is famous air photographs of Palau. How many of you have actually visited Palau? I mean, it's just a mind-blowingly beautiful place. It has, you know, the jellyfish lagoons and other things. But as you get down to the water surface, just as Heinz depicted in his monograph, the shoreline areas are extruded back. I mean, this little thing there on that end is 7 meters measured out. So it's a huge overhang. Something's causing it, and it's presumably these mollusks. There are elevated shorelines. You can actually get sea level things. And notice the elevated shorelines actually have flow stones forming in them. We actually climbed up there 2 years ago with a bunch of Caltech students. We have samples being dated with thorium regression at the moment to see what's going on. But anyway, getting into the interesting story of this micrite, Japanese discovered that it's you can actually dive in and go to the bottom of these little interior lagoons and pull up this mud. And it's very sticky. And in fact, that is my wife, Itsuko, and my youngest son, Koseki. And it is so fun and addictive that I even did it. But there's a little twist in this story because it turns out some Japanese tourists said, oh my God, there's an economic opportunity. And they actually went in illegally and pulled out several cubic meters of this stuff and add tiny bits of it and Palau white. Fun. Okay, fine. Micrite, white clay mud. So anyway, one of the fun projects we did when I took the Caltech students there, we worked with Pat and Lori Collin at the Coral Reef Research Foundation in Koror, and went back to these areas and said, all right, let's see if we can quantify what's going on with these chitons. And when we started looking carefully, we noticed that damn near every chiton was burrowing into these tunnels and leaving these beautiful white fecal pellets. Interesting. Well, you know, we could sample those and so on. And I had been interested in the chitin teeth anyway, because 40 years ago, basically what was my master's thesis, we finally published on the mineralization and magnetization of chitin teeth. And one of the questions was, well, how much could biological magnetite be contributing to the magnetism of these sediments? Same things that Donna's worked on and Bob and I worked on. But we went a little further. In the years since then, we had developed better techniques, one of which was a way to measure clumping. If we start with a magnetic bacterium, and I'll get to more of those there, and then slowly disaggregate them, they go into clumps, we have a variety of magnetic measurements that determine clumpiness. One of them is this ARM acquisition and hysteretic remnants. And so, okay, we've got, you know, the chitin teeth, we have the poo, let's calibrate it. And on these curves, I'm going to talk about the upper one. This is the increase in magnetization as you start with the demagnetized sample and slowly magnetize it. And then you do the backwards demagnetization, so the arrow up and arrow down shows the way it's gaining. And it turns out, and the bottom one is this clumpiness measurement that I talked about. Well, since we had the chitin poo and the chitin teeth, we could actually calibrate it, and that red arrow shows the difference in those curves. okay? So it's actually very nice. What it means is when the chitons are sitting there scraping off the carbonate, their teeth are being ground up, and instead of being very densely interacting like down here, they have tiny little particles that get wafted out. And those tiny particles are perfect for recording their magnetization in sediments. And so, okay, we compared that with the muds from the Palau white mudstones, and it turns out the coercivity spectra is what we call that mud, is precisely that of the ground up chiton teeth. It's just beautiful. And so the conclusion is that white mud is most likely abraded, the coercivity spectra is the abraded pellets. So the bottom line conclusion is that Palau white is most likely chiton species. Okay, and so to go back to Bob Ginsburg, so what? Why is this important? Well, on the one hand, it means it's actually the first example of sedimentary magnetization being produced by an animal. Okay, usually when you talk about biological magnetite going to sediments, it's the well-known magnetofossils that are everywhere and everyone. But here, I think we've got an example where an actual animal is making that. So it blows my mind, it's fun. It could also be why many shallow water carbonates in the lagoons of this environment could be good recorders of the magnetic field, maybe even some of the stuff Don McNeill worked on with Ginsburg and I. And again, yeah, that was this famous seminal paper from Geology 1988, McNeill, Ginsburg, Robin Cheng, and myself. It was fun. And that, so it's another potential source. And of course, Japanese cosmetic companies are selling. Actually, the Palauan government is very interested in that because they're worried about the companies going in and illegally exporting this mud. They don't know how much is there. If it turns out it is the mud from the chitons abrading these shorelines, there might be a limit to that. So getting back to this, the last 10 minutes here, I'll talk about magnetite. The biological magnetite from the chitons was the first one that was discovered, but we now know that there are these wonderful organisms called magnetotactic bacteria. And you can make them go back and forth and turn the magnetic field. It's just fun. So there are half a dozen major labs in the world working on this. They produce chains of magnetite, and this is what people thought were making most shallow water sediments magnetic. And in fact, the more we look, the more environments we find. In fact, the AGU had a session last month where basic conclusion was damn near every shallow water sedimentary environment is being magnetized by biological magnetites, magnetosomes. So it's fun. We also have a good idea of the genetics. They're beautiful for magnetics. It's ferromagnetic. It doesn't need to be magnetized, et cetera, and they're tiny. The other thing that we now know from the work in the last few years, it looks like the trait of magnetite biomineralization is most likely ancestral to the entire bacterial domain. That has to put it back into Archaean time. We published a paper last year in the PNAS on this, and the argument is compelling. Turns out the genes that make magnetite in these bacteria are extraordinarily highly conserved. They follow the nuclear genes, no evidence of lateral transfer. And in fact, you can rule out lateral transfer because there's a whole block of the chromosome that is inverted in the phylum Nitrospirae and not in the other phylums. And that means it had to diverge shortly after the separation of those bacterial phyla. And you do any type of analysis you want to do, there are now 5 phyla that are magnetic. It pushes it back into the Archaean. So we had to have, and the nice thing about that, it's a biological constraint on the duration and the existence of the geodynamo. And I did a back of the envelope calculation paper 30 odd years ago showing that you need at least 6 microtesla compared to roughly 50 microtesla now for the geomagnetic field, to maintain magnetotaxis and to select for it. So that's a biological constraint on the core. Fun. Anyway, and if you put it on the tree of life, the magnetite's got to be somewhere down here. And it radiated out. And those groups that don't, it's very easy to lose magnetotaxis, okay? It's not easy to re-evolve it. In fact, there's no evidence for that. So in fact, most of the bacteria that aren't magnetic seem to have evolved from organisms that were. The other interesting thing is when you think about it, the mud sediment boundary where these organisms live is probably the most stable microenvironment that Earth has ever seen. It's not surprising that these bugs that evolved for that would survive over geological time, even during snowballs, if there's any superficial water at the mouth. So anyway, and I'm even pushing it, I think maybe magnetite aided the origin of life. So, but question about this other domain. Turns out we know that this is all the eukaryotes. We don't know of any archaea, but the eukaryotes, obviously we have magnetite. There are magnetic protists, single-celled eukaryotic organisms that make copious numbers of perfectly sculpted magnetosomes, bullet-shaped things. There are magnetofossils that are monsters. This is the magneto monsters from the Permian, Paleocene-Eocene thermal maximum of New Jersey. We discovered those about a decade ago. And they're really bizarre. They're actually sculpted, single crystal uniform orientations with, over and over again, little like spearheads, like this little microbe is sitting there attacking something. And they actually are found occasionally in balls with them radiating outwards with a hole in the middle. And of course, the Chitons, I already talked about those, they're beautiful. But so what? Well, it turns out we now know that that magnetic material is the magnetic sense organ in animals, higher animals, neurophysiology. And that's putting my biology hat on. All of these organisms here are just a tip of the iceberg of organisms that we now know are magnetic. About 30 years ago, we managed to extract magnetosome chains that are the spitting image of bacterial chains from the nasal cavity of fish, particularly salmon, tuna, and human brains. So, I mean, it's also present in human neurophysiology. The evidence that animals are using the magnetic field was very controversial 40 years ago, but there are hundreds of studies now showing consistency. This is one of the classic ones. You take a homing pigeon, this is just a normal homing pigeon, release it on a magnetic anomaly, and on a sunny day, magnetic anomaly, they don't know where they are. If you release them on a flat area, they go home. Something about 6% variation in the geomagnetic field screws them up. Cetaceans, marine magnetic lineations, again, about 30 years ago, we worried about this quite a bit. We know the classic Vine-Matthews Morley hypothesis. Well, it turns out if you look at where mass stranding events of live whales happen, it's at magnetic extremities of the total magnetic field vector. This was an issue of Science and Engineering, but a paper in the Journal of Experimental Biology of Jim Westphal. The red dots here are mass stranding events, live strandings. And if you just plot from Florida up through Cape Hatteras and onward, showing where the mass strandings are, they're at extremium points in the total magnetic field intensity. Which means, if you think about it, the whales are following the marine magnetic anomalies as navigational cues. Beautiful thing to do. Salmon can use it under ice. I mean, whatever, you can use it at night. It's fun. We also know that this magnetic sense is based on magnetite. And there's a simple experiment that we did about 10 years ago with bats. Dick Holland was a postdoc at Princeton at the time, and he had a beautiful assay for bat homing. And you could actually take the control bats, the bats 20 miles north of Princeton, release them, and they would fly south nicely to the campus. They released them in the middle of a big golf course. You can pulse remagnetize magnetite and flip its polarity. If you pulse it parallel, it doesn't flip. If you pulse antiparallel, it does flip. Well, those bats, if you pulse them parallel, they fly home normally. When you anti-parallel magnetize them, so that you actually remagnetize the magnetite, they go crazy. Boop. Okay, sorry, Bat. They eventually get home, but I mean, we tagged them. But the bottom line is, we have very good evidence that magnetite is also basal to the eukaryotes, most from these things. And the distribution goes from plants to damn near every major group of animal. And it's a basal eukaryotic feature. And so finally, in the last 5 minutes I've got, I'll talk about humans. It's a topic I've been worried about for a long time. Do humans? So I told you it'd be popping up. Anyway, and so again, is there another addition to this? And just for Paul, I put this slide in. Adam Malouf actually did a lot of his magnetic work in my lab. And actually, there was a period of time where every student that came through, we tested. And some of them did extremely well. This is a two-choice paradigm. When you start getting strings of many, many in a row with 9 chance in a billion probabilities, you say something's wrong. But then the next day they come in and they sneeze and they get randomness. And so we didn't have control of the behavior. About 4 years ago, we got funding from the Human Frontier Science Program in Strasbourg with a colleague of mine, Shin Shimojo, who's the Gertrude Baltimore Professor of Human Psychology. And he said, you're stupid to do behavior. Just look at the brain waves. If humans have a magnetic sense, we might not be paying attention to it. My students don't pay attention anyway. And so that led to a completely different twist. And in fact, if you want to read about it, if you Google Kirschvink and bioarchives, the manuscript, which is currently in review for a journal from the American Society of Neurobiology, is online and you can look at it, but just don't tell reporters about it yet. Anyway, the idea is to monitor the brain waves in a controlled room where we can silently move the magnetic field subtly, putting every control we've ever developed for animal navigation experiments in it. This is the real example, the Faraday cage that was being built, the subjects in there, double-blind controls, control flux gates, et cetera, et cetera. Subjects, yes, we do inject their brain with that's conductive jelly on the skull cap, 64 leads. And the basic idea that we came up with, turns out the brain waves are, you can put electrodes in the brain and monitor voltages, but there are some fundamental patterns in the brain wave. One of those is the alpha wave. And if you're sitting and thinking meditating in a chamber, thinking about clouds and not worrying about anything else, the brain likes to be in sync. The alpha wave, 10 hertz band, it comes up in power. Now, the alpha waves go up and down and up and down. You can see, if you look along this thing here, there are some intervals where the 10 hertz signal is good. These are 1 second ticks. It goes up and down and up and down. That gives you a peak there. It turns out the neurobiologists have known for about 30 years the amplitude of that alpha wave signal depends upon sensory input. If you're sitting there in the dark, you get big alpha waves. If I do something weird, if I flash a light, give you a sensory signal, the brain receives that. Parts of the brain that are in this hum have to go away and do something else to figure out, is it danger, is it not? And those alpha waves collapse. So you can use the alpha wave to test for sensory perception and transduction. So 2 major results. If you shift the magnetic field up and down, the brain waves collapse a few seconds later. If you rotate it counterclockwise down, the brain waves collapse. And it's kind of like this. The strong alpha, it's called an event-related desynchronization, or the counterclockwise down. So here's an example of that. This is a time frequency. Again, the power spectrum. Blue is very low power, so the alpha wave collapsing gives you blue on this. Strong alpha waves would be red. And so if you put the magnetic signal here, the field rotation, the brain, hey, and think about it. Ever since you were a fetus, if your mother moved or you moved your head, the magnetic field is moving relative to your skull, but your ears know it, your somatosensory system, your brain is telling this. So you know, it's no big deal. I move my head, the magnetic field moves relative to the head. In our experiment, we moved the magnetic field, but the brain didn't tell it to move. So what the hell is the magnetic field moving? Brain freaks out. That's what's happening here, okay? And so you can actually see that, and you can actually parametrically do statistics, et cetera, et cetera. So here's an electrical, and so the question is, the brain is doing this, but what is the mechanism, okay? Is it electrical induction? Is it magnetite? Is it a quantum compass? What is it? Simple test for electrical induction. We have this counterclockwise response that's extremely robust. So let's compare that rotation like this, with a rotation like that. dB/dt is identical, okay? Horizontal rotating component, static vertical, either down or up. And here's the difference. Huge response downwards, as in the northern hemisphere, no response in upwards. In fact, in the next slide, we'll actually show you this. This is now brain head looking down. These are the nose, ears, and the alpha band power is in this red to blue scale with green. The upwards component are the top rack, downwards the bottom rack. This is counterclockwise, first column, clockwise, second column, fixed control. We pretend to move it and analyze it as if we had moved it, but didn't move it. So watch what happens. Here we go. Magnetic field change there. See an alpha effect? I'll do it again. Yeah. So watch this lower left panel here. Control, magnetic field change, 1/10 of a second, brain waves collapse over it. Not the up one. So the top and bottom have the same dB/dt, but different result. It's not electrical induction. It's not induction in the wires, in the artifacts. That is actually an extremely powerful result. Anyway, there's also a quantum compass test, very similar thing. Quantum compass, as many quantum biologists have proposed, actually has an interesting limit. It can't tell polarity, it can tell axis. So it can't tell that from that. So down to the north cannot be distinguished from up to the south, and that's those two things. And so here's this test, okay? Here's the direct test of the quantum compass. Again, magnetic field change there. The different subject, a little longer in the brain response, but boom, you get it down to the lower left, and not the upper right, upper left. So, and so anyway, I'm more or less out of time. Let me, but anyway, we know there's massive cells. I'm almost done. What I would say then is that this magnetic biosphere, we probably got the magnetite in the endosymbiotic event that made the eukaryotic cell, and it's radiated ever since, and we're certainly part of that. So conclusions, magnetite from the chitin teeth in some situations can actually be an important magnetization of marine sediments. Earth's magnetic sphere is magnetic, and da-di-da-di-da. Humans are part of that magnetic biosphere. So anyway, there's our team, and we actually now have major funding from DARPA. They're interested in all sorts of things about humans being able to navigate and so forth. Thank you very much. And again, kudos to Bob Ginsburg. All right. DARPA, Defense Advanced Project Research Agency. Yeah.
Joe, what do these tiny particles in your beautiful facial cream, what do they look like?
That facial cream, if you actually read the fine print in Japanese, it's homeopathy. They dilute it and they dilute it and dilute it. A little bit of touch from it. In the actual muds, we haven't extracted them. But the rock magnetics is pretty clear. They can't be particles more than about half a micron in size. compared to what the previous one is. If there were more than that, then the clumping would be way down. Yeah. Oh, well, obviously, those pellets will get washed out, they'll sink, they'll disaggregate, the bacteria will leak the slime off of it. Yeah, but I mean, those particles are so white, you can actually feel the fine-grained micrite mud in them. So I think it's pretty clear. That is the source of the stuff right offshore from them. Oh, no, the teeth are ground up. Yeah, well, the teeth are in the radula. And when you actually look at at the chitin radula going in, you see that the most mature teeth are abraded, actually, down to nubs. And in Lawenstam's tanks, when I was an undergraduate, one of the things we reported in that 1979 paper was the rate that the teeth were formed. It's about 1 tooth per day per animal. Yeah, that gets done. And that's the replacement in the cryptochitin. And we did that by magnetizing the chitin with a big magnet, waiting a month, and then dissecting tooth by tooth out with non-magnetic tweezers and individually measuring them in the squid magnetometer. And you can actually see the teeth that had been formed before we hit it with a magnet were strongly magnetic. The new teeth were very weakly magnetic, and there were about 15 new rows that were not magnetic in most of the radula that we looked at. Well, in that particular experiment, we were not cueing them in any way. They're passively sitting, trying not to fall asleep for 7 minutes while we do it. The computer randomized the trials, the length and durations. Nobody knew what was going on except the computer. But of course, the Fluxgate and the EGs were all recording the data, so we have a complete record of that. And then we go back and process the data. Those maps are actually based on about 100 rotations each way. And you rotate it and monitor the brain wave in the 3 seconds later, and then you rotate it back randomly, and then you go back through the data. We have 50 gigabytes of data that are deposited online at the moment. Anybody can go through it and play with it. You're welcome to it. So, yeah. So there's no consciousness in that. And in fact, more recently, we put the push button back in. Same experiment, except 3 seconds after the field is shifted, the subject hears a tone, and they have to guess whether or not the field changed. Those responses are completely random. However, if you synchronize the brainwave pattern to the actual contact of the push button, there's a huge magnetic effect just before it pushes. So something in the brain is saying yes or no, but it's not getting into consciousness. It's there, but it's not conscious. Oh, and one other experiment. We did the bat experiment on one subject. We converted the downward response into an upward response. And then the IRB won't let us do that experiment anymore because it caused the subject's eyebrow to twitch. I kid you not. Yeah.
Okay, last question. What grinds the magnetite when you're working on particle speed calcite or
Oh, yeah. It's actually, magnetite crystals are held in a matrix of collagen fibers. And it's the collagen that gradually breaks away. On the other hand, the structure of those teeth are actually, it's about a micron size hexagonal wall of major collagen that comes down. And then each 1/10 of a micron crystal is held together by a little layer of collagen. And those layers slide gradually when they go across, giving it extra strength. If it were a solid crystal, it would fracture and break. But by building it, you get an incredibly hard material. And that's what the material scientists are so fascinated with. That biology is converged on a structure that makes incredibly hard materials. Harder than the magnetite would be itself. But it will wear away gradually, just like sandpaper does. Yep. And that's what we pick up in the magnetic signature. Okay. Fun. And if I could have my
So when he was here, he built the petrophysics lab, and we did a lot of experiments on looking at sonic velocity, porosity in carbonates. And he went out to build quite a bit of Ginsburg. And later he went back to more environmental geology and in lakes, and he works in Bern now, the University of Bern. And he will give us the comparative sedimentology approach in Lacustrine environment.
Thanks, Gregor. What a nice event, and what a pleasure and honor for me to witness this and to be invited. Thanks a lot to the conveners. And I came here in 1990, 1991, around this time. And what better can happen to a guy from fresh graduated on a Swiss university to get to this environment, to get to the CSL, you know, to the aura of Bob, to get inspired by him. And actually, it's quite what Noah said. You know, you came from Wisconsin. You know, you came in. I think we both make good cheese. You know, so it's kind of it's maybe we have the similar experience. You see, look at this. So I had the chance to get to the Andrews Flat. Thanks, Eberhard, for making these pictures on our field trip. I get back to the field trip later. But the fact of getting inspired into a certain way, also directed by Bob Ginsburg, was fabulous. And Gregor said I'm more or less the grandchild, so Gregor was my real supervisor, and I think like this I could probably get the best out of both worlds. So I think we heard that Ginsburg was a really harsh advisor. With me, he was not like that. He trusted Gregor that he does the day-to-day supervision, but of course I got then adopted by him and inspired and directed, although I don't pull the boat exactly in the same direction as he points to. I shall see. Okay, when I came was in the 90s actually. The first time I came then was at the start of the Bahamas drilling project. What an experience. And it was also the reason why I'm not part of the Fisher Island gang. So I only spent 1 or 2 nights there, and then we left, and then the island was sold, I think, 3 weeks later. So that was the beginning here, and then imagine the innocent Swiss graduates being dropped in this drilling gang here consisting of Louisiana and New Jersey drillers. That was really an experience which I never will forget, and I think it formed me in a way. I think it was the best years of my life, and it still influences me today. And this is something actually what I want to talk today about. I'm not going to present the stuff, the projects I conducted at that time, but I want to kind of show how the CSL lab, how it inspired me with approaches, with way of thinking to do my current research. And you'll see some parallels there, that we always get inspired by the CSL way of thinking, Gregor and all colleagues, but of course by Bob Ginsburg's inspiration as well. I will do this twofold. In the first block, we go a bit away from carbonates. We look at extreme climate events as natural hazards in the first section. And in the second part, we go a little bit back to carbonate platforms. So these are kind of 2 parts. So let's start with natural hazard. And I come from Bern, and that's an image of Bern 13 years ago. We have major floodings, climate extremes, and Bern has a history of floods. That's the Old Town, the lower Old Town. It also has a history of showing what you do with people who cause this natural hazard. So that's a witch burn event in Bern. Good 400 years ago, and it just shows the significance why we need to understand natural hazards. And we as geologists, we have an important role in there. So we have the geologic record that can actually tell the history of natural hazards and the frequency, the intensity, and also the causes. And this is our almost societal role. But who am I to tell you here in Florida? Because you have hurricanes, and I was almost here at Hurricane Andrew. Actually, I was with Peter Homewood in the Canadian Rockies. We got snow in August in our field camp, And 2 days later, Hurricane Andrew hit, and I returned, I think, 3 days after hurricane. And of course, when I came back, and once the National Guard kind of left the county again, and everything went back to normal, a few weeks after that, of course, Bob Ginsburg came and say the classic Friday afternoon tour of the hallways when he said, "Who's coming up for a coral reef visit tomorrow?" And then so we could go out on his boat, and after Hurricane Andrew, his idea was to track really the effect of Andrew on the sedimentological record. And he had this idea, if I remember right, that the big blocks of coral reef were kind of torn off by the storm, and especially those who had this gorgonia hanging on them so that they could act as floats or as sails. I don't remember honestly whether this first trip to Bachy Shoal was successful, but of course, if you translate it to a bigger context and you look at all the storm tracks of the last decades, of course the geologic records are critical in order to do regional records of extreme events. And Bob Ginsburg did this already before, 1 year before Hurricane Andrew hit in 2001. Actually, some of us here in the room, Eberhard amongst others, with Andy Reesey, John Gopol, Jeroen Kenter, we went to the Andros tidal flats with the idea to see the effect of hurricanes in general, to see the layers on the banks, on the levee deposits around the tidal channels. And the interesting thing with this trip was, you might notice a bit weird clothing that we are wearing. And we've heard that Bob Ginsburg was very aware of the costs he would spend for such a field trip, and actually he said, we need long sleeve shirts and we need all this material. So he sent Jeroen Kenter a week before the field trip out to some used clothes stores, and where you can get the piece for $1 or less. And he came back with these designer clothes. I mean, these were all kind of Gucci and Armani. Look at my pants. I have a white shirt from the finest. So we were this weird-looking, well-dressed team that went into the field, and it was one of these inspirations. It was great fun, and actually we did a series of courses. We took a series of courses in the tidal flats, and it was known from previous studies. There are some iconic work from the 60s and 70s that could show that these layers do consist actually of mud. It's not sand, so we could confirm this here. But there are some nice, which we interpreted also as storm layers. But one of the findings of this field trip actually what that is. So if you compare the course we took, let's say from one tidal channel levee to the next one, it turned out that we could not correlate these layers. Partly we could do, partly we could not do it. So in order, what I said, to do these geologic reconstructions of hurricane frequency, it's a very patchy record, so maybe not that well suited to do this task. And I just want to add here the post-Andrew cruise that was done then by Gregor and Mitch. You were and Gregor gave me this picture. You then saw the layer that was produced by Hurricane Andrew. Here we see already the laminated crust on top that formed, I guess, a few years later. But here Andrew left this nice piece of a nice white mud layer. So basically, this is the approach which I got from Bob and from the CSL lab, and it's kind of the comparative from the actual event, and then you want to see how does it look in the past. And so now let's translate this. So then natural hazard in Europe are mostly dominated by, we don't have the real hurricanes, but we have strong rainstorms, so we have big floods as well. And we were 2 years ago, I was with my students in the Swiss Alps. We had a mapping course, mapping Mesozoic carbonates, Mesozoic siliciclastics also. And after a long day, we sat on the terrace here of this hotel, and we're watching kind of the 100-year event of rainstorms evolve. It was kind of a coincidence. We had actually to leave early because we saw on our radar, we saw it coming. And actually, that's how it looked like. So that was the view from this terrace. We had the beer in our hands, so to speak, so we could look. All the creeks left their beds. We saw here the red flood overs. We see all the slopes getting mobile. We had slope instabilities, and it was quite a dramatic event. And we were happy that, I was especially happy as responsible that we got all students back. You also see this crazy guy. It's kind of a hurricane evacuation equivalent. And you see here all the red water masses pouring into the lake, and what the hell is he doing? So we expected that actually these waters would take the boat anytime into the lake. He succeeds, so he gets through. But you see a little bit of difference. So that's kind of the hurricane evacuation from Andrew, which is a bit more more controlled, but you have more people. So I guess you can't have everything in life all the time. So, but again, juxtaposed here how you handle these natural hazards. So at the same time this happened, we of course thought, well, we have a nice record of precipitation. So that's the radar image of cumulated rainfall of this event. And of course, while having that beer and looking at this flood, this big event evolving, and especially then the next day, by looking kind of at the scars it left in the landscape, we thought, my gosh, all this plastic material that must leave their traces in the basinal record of that lake. And in addition, these are continental Permian and Triassic deposits which become eroded. That must be really a nice contrast in the lake sediments. And then similar to Bob's rapid response event after Andrew, we said, okay, let's see whether we really find this event, and let's see whether we can do a flood chronology over a longer time period. And then so the idea is that, of course, the flood makes a nice underflow turbidite, and we should see a clastic layer in the basin. Okay, so then we apply the usual. 2 years later was Nicole Glaus who did her master thesis with this, a nice little research project. So we, similar as different lake, but it's the same concept. So we shoot seismic sections, so we know exactly where we have to go to core these records. Then we did short coring from the ice in the winter. We take long cores, 8-meter long cores from drilling platform in summer. And sure enough, we found these. We found the layers, the red layers that we expected. So here you see the sediment-water interface. That's just green foam on top. And you see here a nicely graded layer with the coarse grain size. Maybe you can see it. And a fine clay cap on top, the classic graded layer representing a flood event, and that's the 2016 flood event. And what you also can see sometimes is that you have kind of an inverse grading reflecting the waxing and waning of the flood. So it's kind of like a hydrograph if you look at the grain size distribution. We also were happy to see that it's not the only layer that occurs in that section, but that we have a whole series. And moreover, we can actually correlate them from across the transect, across the lake. And that's kind of in contrast to what our experience on the Andros tidal flat told us. So we are happy. So we had so Nicole could actually lay a good base to do a classic flood reconstruction there, and that's the whole record. So we have 8 meters. She has about 144 flood layers. You can characterize them with different proxies. You can look at the grain size or at the density. So basically, in the end, you can define a flood record, which then you only need actually to date. And for this, we take just the classic radiocarbon chronology by subtracting the event as rapid layers, and then you get a dated flood chronology for all the 144 events. So how does this flood record look like? So you notice that actually we do have a lot of flood layers here, and that's about 4,000 years BP. And we have almost no flood layers prior to that. And here down we're already in the late glacial, and we have a different sedimentation system in this basin. So if you look at this record from 10,000 years to today, we have this almost lack of flood layers, and then we have an increase, but we see nice variations. So there were periods when we had more flood layers, and there were periods when obviously the climate was in a way that there were less flood events, and this is what we wanted to go for. We also could, because we quantified the whole basin, so we could quantify soil erosion. We could say how much soil was eroded per year in tons per square kilometer into the basin, and how much did the particular event, the flood event, contribute to this thing. And we see a dramatic change in 4,000 years. And of course, then we said, well, you know, our goal was a paleoclimatologic reconstruction. And it can't be that the climate at 4,000 years shifted so dramatically. And in fact, in that particular catchment, 4,000 years marks most likely the onset of alpine farming. That brings us back to the cheese So we have somehow another effect in there, which actually we want to avoid in this case. Of course, one can always turn it around and look at the advantages of that too. But in order to get the climate to kind of subtract kind of farming effects, or also recent effects from ski slope for the last 50 or 100 years. We did an extensive study over the entire Alps, north and south. We looked at about 2,000 lakes of various elevations, various sizes, in order to really get the over-regional signal for such a flood reconstructions. And that's then kind of the database. So these are the lakes north of the Alps. It's always 10,000 years, so we have kind of here a good dozen individual flood records. And in the end, we basically merged them all in one, actually in two, one for the north side of the Alps and from the south side of the Alps. And the motivation behind that is that you think it's controlled by atmospheric circulation patterns. If we have the moisture coming from the Mediterranean, we get especially the southern lakes that react. If we have the westerlies coming in, it should be the northern lakes. So that's then the 10,000 years of flood history from the north, this one from the south. We see periods of increased flood occurrence. I don't want to go into these details. I don't have time. I just want to look at the northern Alps in the last 2,500 years. So what do we see there? And this is the last 2,500 years of flood history north of the Alps. We look at a good 800 dated flood layers. And these are all these layers. You see each lake is different, but we can merge a nice signal. We get here 2,500 years of flood frequency. And then we said, okay, let's compare this now with climate. So for instance, with temperature. And we had a totally independent temperature reconstructions. That's from tree rings nearby. So there was a tree ring study who made a nice temperature reconstructions. And then we have an incredible match, basically all of this. So we have this kind of centennial fluctuation. Basically every single peak actually did match this thing. And the only thing that we had to watch out is that we had to turn around the temperature. So we had an increased flood frequency at cooler temperature. And that was a bit problematic because suddenly all the climate skeptics came and says, you see, climate warming doesn't mean automatically that you get more floods, that the natural hazards increase. So we said, well, be careful. So we don't look at intensities, we look at the regional signal, and we need to explain somehow why this occurs. Because if you use Clausius-Clapeyron equation, with warmer temperature, you can transport more humidity. You should get an increase in flood frequency. So actually we talked to the weather people, to the climatologists, and actually the explanation is fairly simple. So if you have a warm summer, all the Atlantic fronts actually go rather north to Scandinavia. And if you have a cold summer, you have a different NAO index, and you have the fronts, the westerly, reach straight into the Central Alps. And like this, you have this dependence on temperature and climate. And we really have to raise the warning flag there that one cannot expand this to a global statement that if we have a global warming, that we get less flood frequency. But on a regional time scale, regional spatial scale, one has just to be careful by always kind of alarming that every natural hazard will increase. But this I just wanted to show us kind of while kind of Bob Ginsburg was watching Hurricane Andrew going over it, having this idea of checking. We had when I sat there on this lake 2 years ago, I had the same feeling. So look at this material. This is a good story. We have a unique chance to calibrate our system and to do kind of a comparative sedimentology, also in a different area, and maybe not with the classic carbonate that it was originally intended for. Okay, let me get to the second example. And that is kind of a comparative sedimentology that is not necessarily meant from the modern to the ancient, but that we can just take two different systems and compare them directly. In this case, I want to compare marine systems with lacustrine systems. And I want to do this exercise here in the Swiss Jura Mountains. That's a master student and a scientist who works with Raphael Meyer and Stefano Fabri, who studied this basin. This is the Lac de Joux basin. And if you look from space, of course, a lake basin, it's like an ocean from space. You don't see a lot. So actually, we do operate our multi-beam bathymetric systems. Of course, we are geologists, sedimentologists. We want to see the basin. So we do these high-resolution bathymetric surveys. And then after that, the lake looks like that. And it's much more interesting for us to start the processes in there. In fact, what we were looking for were, for instance, fault lines like this. So it's an active area, so we see some fault lines, we see some mass movements, and we see, for instance, here, this like a bullseye in the middle of the lake. That's a beautiful subaquatic spring, which caught our eye. So we have from the car system board popping up like a pockmark or something. So that was our original goal. But when I looked a little bit closer to the map, I saw in this area, I saw things which I said, what is this? So I zoom in a little bit. So we see in red colors all the water depths, let's say maximum 6-7 meters. So these are zones, these are like banks which are totally flat on top and which basically were built up to lake level. And of course I said, whoa, this is bizarre in a lake system. So how can we do this? You know, it's not so easy to do. And kind of then my comparative mind, my deformed comparative sedimentologic mind started to spin. And I was inspired, so I spin first a little bit the map. And anybody has some connections when he looks at this image? Yeah, we have kind of a coast, a lagoon, then an edge, and then kind of these isolated things. Belize, huh? I mean, it looks just a little bit off scale, but it looks one-to-one like Belize. Can it be that these are carbonate buildups which actually grow up to lake level? And then so to visualize that, we can also look at that. It doesn't show up, but on some of the satellite images you would see here the shallow water zone. So they do have this suspicious bright colored sediment. And to verify that, let's go back, and we took a short cohere and we look at the seismic section there. And I must say that's a waste product from this survey, so because that's the only example I found, because we didn't aim for this, but it's in our neighborhood, so we can go back. Okay, let's look at the first at the core, and we know this color, huh? Beautiful. It's very carbonate, with 80% carbonate. Mud waxstone, what you don't see, it has a lot of little bivalves in there. It has ostracods. It has a lot of carbonate producer. So in fact, these shallow banks are the place where this lake produces carbonates, and that's what we call somehow Lacustrine chalk, or in German we say Seekreide. And it seems like that builds up nicely to maybe to wave base, to the accommodation space. So sure enough, we then also evaluated the seismic section that goes across it, and here we see a seismic section going across this bank. So this is where the core came from. We have this flat-topped bank. We have a slope there. And then what was somehow surprising, that is, if you look closely, you see that, whoa, it seems to have a core here. So there's something in this bank. It's not just layer cake. So there's something on one side on this bank. And so the question first is, what do we have in this bank? And if you look a bit at the geomorphology, I need to mention that this is a glacial lake, so that has been formed after the glacial retreated. So it's a glacial over-deepened basin, and that's basically the glacial flow direction. So I think we see a lot of glacial, subglacial morphology here, and in fact this we interpret as a moraine, or basically or an esker, or the basal lodge matilla, or something that was left over from the glaciation. If you then look at this, you see that you have a series of inclined reflections here. Also, these are inclined. So that bank did not build up vertically from below. And of course, I mean, we've seen the line of the Bahama Bank now numerous times today. The really milestone paper by Gregor and by Bob Ginsburg, that like the Great Bahama Bank, which appears totally flat, doesn't consist of a layer cake. So it has some parts which are kind of the core, which were aggrading at one point, and then the bank started to prograde over, and that edge moved out. And when you think about this, we might, you see, this is all new, we haven't looked at this in detail, but we might have a very similar process here, that in fact we do have kind of uninherited topography that's left by the glacier that comes in a productive environment. So it's the karst mountains of the Jura. We have calcium saturation on carbonates in the water. We have warm summers, so we can precipitate carbonate. So we might have a nice analog to the carbonate bank, which I started to study in the 1990s, and I didn't forget it obviously, because it immediately struck my eye. And to spin this idea a little bit further, so the findings summary, we can say, well, these are flat top. They are made of carbonates. They nucleate on structural heights, and they seem to prograde towards the northwest. something. I don't understand it because that's not the main wind direction. So that's something we have to look what controls this. But let's look also a bit closer. So what we also see that we have like straits in between, and then came around, well, maybe if we have enough time to wait, we might get something like a coalescence of the bank. So maybe if we wait a couple of decades or 100 years, this strait closes, and the analog situation to the Strait of Andros might pop up here, that we also find in other of these shallow water banks actually some deep water straits in between that now are covered by a flat bank. Of course, deep water here means maybe you talk of 10, 15, 20 meters in between these banks. But it's driven by carbonate production, dependent on the environmental condition of the water body. That's an important thing. So carbonates get back in my life, which I'm happy of, and I want to finish with one example from the biggest lake in Central Europe, on Lake Constance. We also have here spectacular bathymetric data. We have here a meandering channel from the Rhine inlet all the way down. But I want to look here on the southern shore, on the southern shore of Lake Constance. We zoom in because the topic is the shallow water banks, and this is also a lake that produces its own carbonate. And we did the survey, and then we saw here the kind of a shelf edge. It goes down. This is about 50 meters deep here. The orange is land. So we did this we combined the bathymetric data with green laser LiDAR technology to map the shallow water zone to 5 meters, and this also gives us the land. These things, we have a lot of pock marks. We have most likely some iceberg scours here too, so that's the glacial history. But if we zoom in this area, we see something really peculiar in about 7 or 8 meters of water depth. So there we had, in the middle of this bank, this almost super regular series of mounds. These mounds are maybe 2-3 meters high. They are up to 15 or I think 20 meter diameter. And then the speculations, so we published that data, and then the speculations went wild. What is it, So, and some of the speculations were clearly towards a human origin. It's, I mean, if I look at it again, I think I'll tell you that actually it's most likely that it is of human origin. We don't know how old it is. We don't know what it is and what it was meant for, but we wanted to check a bit in which setting, how does it work? So divers found out that they were unexplored. They consist of kind of blocks like this. So these are piles of rocks. And one guy said it's a glacial moraine that pops out, but we said, well, with this equal spacing, that can't be. So also let's look at some geophysical data. So a colleague, Jens Hornung, from University of Darmstadt, he actually made a ground-penetrating radar survey. He has a very cool sled which he pulls across the lake floor, and he could make a line there. And that's what he could show. So that's the shallow water bank from kind of the coast to the shelf edge would be here. I zoom in on this package here, and this is the mound. And this is the bedrock that dives down. And here you see beautiful, this carbonate sediment, how it basically progrades out into Lake Constance. It's different than before, so this is it's much stronger currents there. So it's a current system that transports the carbonates which you produce there, transported out. And you see that actually the mounds are embedded in this prograding package. So let's compare it with a radar line in between those two mounds. So we just juxtapose it to this. So this is the same shelf, so to speak, the same carbonate shelf, but in between two mounds. So there we don't see a mound that was built. But, and now comes the interesting thing, we see the same amplitude anomaly in this prograding succession. So we can do somehow a sequence analysis, track some reflections. We have some geometrical features here, but we see that basically this mound, which we think is really made by humans, maybe in the Neolithic age, that it's really tied somehow to this amplitude anomaly here, that is somehow in the middle of the entire propagation as we see it today. So I cannot give you a definite answer, but what our current working hypothesis is that kind of the easiest way to make here a amplitude anomaly in the GPR data is that you have lake level changes, that you bring down lake level, that you change lithology, that maybe you stop carbonate production in this part, and that you allow the people to move out, and for whatever reason, pile up these rock mounds along the shore. And we talk about, I think it's 60, 70, 80 mounds. And I can't tell you how old it is. The ongoing archaeologists think it must be somehow Neolithic. That would be maybe 5,000 years old. But we start now a project that we do actually a drilling transect here, that we want to date this progradation. And this is all new. So these mounds have never been seen before we made that survey. And the timing of this shallow bank progradation into the deep basin of Lake Constance has also never been dated or shown. But again, so coming from the Bahamas drilling project, coming from doing the seismic surveys on the marine bank, we've seen similar geometries. We can do seismic sequence analysis, and you can get inspired to look at these things, at these elements, which are also part of the carbonate legacy, so to speak, that the CSL and that Robert Ginsburg stands for. Okay, let me conclude here. So we don't know yet the purpose or the exact age, but that's what science makes about. So what you can answer here, that also this will deliver an element also to the human history of the area, which to date is not known. In addition to all the understanding on the paleoenvironmental controls of these carbonate banks along the lake shores, which we then can correlate to climate changes or to other environmental water changes. So basically, I'm at the end. I could convince you a little bit that we can expand a little bit the CSL philosophy to other environments, to lacustrine environments, to extreme events, to natural hazards, as it's done already now here as well. And the last thing I want to mention here is that, next to the big thank you of course, to Bob Ginsburg, but to everybody, to Gregor for bringing me over here to Miami, is that what this special way of thinking that Bob Ginsburg represents is really a way, this questioning, everything what we heard these days, that's something which I, even without noticing sometimes, which I give now away to my students. Huh? So this is something that we can pass on. So these are the grandchildren, so to speak, so that his spirit somehow lives on. And I'm really happy to be part of this chain reaction of doing this modern sedimentology. Thanks a lot. Thank you. Tricky signal, because these lakes, they have a well-studied history of eutrophication. So we had all this nutrients load from agriculture, from human wastewater. So actually that increased by this process, even without the temperature increase, a lot the carbonate production. So we had a much more photosynthetic activity, much more diatoms that bloom. So that's kind of the prime control on the changes in carbonate production modulated on this is this effect, so that we have the kind of the weathering changes in the catchments. But I think they are second order, I would say. So if we would have to go to a really pristine, untouched environment in order to see that, and the Swiss lake do not offer that pristine environment.
Flavio's understanding is able to record the common part of the succession, which is fairly fine grained. What's your expectations if you're able to record the whole succession?
Yeah, that's the plan. So the question was, what if I would go further in the lock tissue, what I would expect. So sooner or later I would hit the glacial till, the basal lodgement till, but that wouldn't be the interest. So of course there you get the point as soon as the glacier lifts, as soon as it lifts its bed, its till, it starts first with a subglacial lake that is clastic, that is mostly clastic. And soon enough when this ice completely vacates the basin, and it's also about the time when climate gets warmer, when the lake warms up, when the biologic productivity kicks in. So then you expect that the carbonate set in. But it's not like, I do not expect like we have it here, that you have a transgression. So that's a total different system. What we see now from Florida Bay is that you get the shell layer at the base or these parts. I think when productivity kicks in, the lake is already at that water depth, maybe even higher. So we don't expect a forcing, but I expect a slow increase in productivity indicated by a gradual increase in carbonate content.
Still fairly fine grained sediment.
Still fine grained because we are 200, 400 meters offshore, and because these are really isolated banks, so we don't expect the delivery of coarse material. The shore is different. So the coastal platform in the last example, that's really coarse, because you have longshore currents that redistribute the clastic material. But they do not reach these isolated platforms out in the water. Okay.
Yeah, how is the medieval climate anomaly, recorded in your storm layer or some flood layer? I was not able to understand.
Which anomaly?
The medieval climate anomaly, between 900 and 1400, where we know that most of the glaciers, in the Alps, melted tremendously.
Yeah.
And I wonder how this would be
Yeah.
translated.
It was really a reduction in flood intensity.
Even the glaciers in the Alps would have melted.
Yeah. Well, we talk. You see, for instance, if we go to the Little Ice Age that follows thereafter, we have actually the highest flood frequencies in the Little Ice Age, and we see if we do spectral analysis, we see the solar frequencies in there. We see the solar minimum as well. So the highest flood frequencies in the Little Ice Age with cold temperature, and then this is the medieval anomaly here, and we see that flood frequencies are really low. So yes, so it was an issue. So people said, you see, climate change is not a problem. But I think there really the climatologists help us kind of. We ask them, so what if we have these summer temperatures? How would the atmospheric circulation pattern see? And they really say that we had, for instance, 2003 a summer which was extremely warm. We had no extreme events as well. So it was so dried up that it was not even the summer thunderstorm convective systems that came up. And so to answer your questions, it's really a low flood frequency when we have these warm and when the glaciers are short.
And these are flooding events, yes. It's not kind of a decade or century.
Yeah. No, we talk of individual events. So this can be totally separated from general precipitation. So this is not a precipitation record. It's the record of extreme events.
Yeah. And so what is the rate of sedimentation that changes within this?
On the long term, not that much. Not that much. We calculate the fluxes, but there is no massive change. Because that would relate it then to the precipitation in general.
Okay, thank you very much.
So I am losing my voice.
It's on this one, right?
Thank you, Gregor. This is my second day in Florida, so already I'm losing my voice, and I'm going to have to resort to the microphone. It's a real honor to be here to represent the AGRRA team in the last talk in this session of the Legacy for Bob. I think his aha moment with regard to reef health occurred during the International Coral Reef Symposium in Guam, when Bob Budemeyer started the conference by saying that climate change was going to further trash the reefs that we were already destroying, and that was followed by Clive Wilkinson saying, well, we'd already destroyed 10% of the world's reefs globally, that another 60% were in various stages of decline, and there was only about 30% in the most remote parts of the Pacific Ocean and other unexplored, virtually unexplored areas in which there was any hope that they might be in good condition. Well, Bob objected strongly. He did not believe it, and he famously said, you know, they're just not looking in the reefs in eastern Indonesia. By that, I think what he meant was that everybody knew the formerly beautiful reefs off of Jakarta in Java had been obliterated basically by development, but that was maybe not representative of Indonesia as a whole, particularly in the less inhabited eastern islands. So the reason that he was so adamantly opposed to this emerging consensus at the time was twofold. One, he said that we biologists were thinking in timescales of decades, and as a geologist, he knew that reefs waxed and waned and waxed back again over timescales of millennia in response to natural climate changes, and we were only looking at a few reefs, mostly located in beautiful locations where we like to go diving, and if they changed in the short term, they could, you know, only get worse. So he came back to Miami, and with Peter Glynn, he organized what was called the Coral Reef Love-In by some of the other geologists the following year, and that was the global aspects of coral reefs' health, hazards, and history. And that was a huge success, and it spawned all kinds of follow-up activities, including the first ever International Year of the Reef in 1997, of which he was a co-chairman, the International Coral Reef Initiative, the Global Coral Reef Monitoring Network, the Coral Reef Task Force of the US government. Reef Check went around the world with volunteers surveying reefs. And there were 62 presentations of case studies, many of which said, my reef is declining. And in one of the panel discussions, the idea was proposed that, well, we'd reached the stage of triage. And really, the reefs of the Florida tract were already in such decline that there was no need to, or compelling reason to commit any further resources to try to conserve them because they were gone anyway. Well, once again, of course, Bob took serious offense to this statement and said, but there are large areas of healthy reef and conservation efforts are still required. And the next year when the proceedings were published, that would have been in 1994, and with Peter Glynn, that was the consensus of the first of the 4 major conclusions that were made. Yes, they're in decline, but we don't really know what's happening globally. But by that time, Bob was already trying to find out, and especially to learn what was happening in the Florida Keys. So he had assembled a large array of helpers, some biologists, some geologists. I've listed the names of the primary ones on the top there. Some of you are here, you'll recognize your name. If you were a co-author on one of the 2 publications that resulted from this work, you're either listed as 1 and or 2, and they're mentioned at the base. And he used, you know, an equally large number of approaches to try to answer the question of how can you rapidly evaluate the condition of reef corals over large areas, because he wanted to get a representative sample that you could put on a map and convince anybody that things are happening regionally, at least, you know, in this part of the world. And And he started with the patch reefs in the Florida Keys and the big, massive corals. Bob always thought big. You may have noticed when he signed his name to something, it was not as large as President Trump's, but it was big. And between 1994 and 95, he made a series of surveys, the numbers are shown on the slide, and concluded that where the corals were not doing well, it was largely the effect of Hurricane Andrew, which had occurred a few years before, as we've heard already. And when he went back and did with a whole new set of methods in 1996 and did another whole series of surveys along the Keys, he decided that the reason that, which was really known, the corals on the reefs in the middle and lower Keys were not looking as healthy as the ones up around the upper Keys, especially out at Carysfort, that was due to a combination of greater exposure to storms in the lower Keys and to the effects of the warm, turbid, saline waters that we've heard can come out from Florida Bay and flow over the reefs in that area. That's the iminical water of Ginsburg and Shin, or Shin and Ginsburg. And I might say that along the course of this, he decided to pay a lot of attention to what is called partial mortality. And the idea there is that as colonial corals get large, they die off in some areas, and these areas tell you what was alive at one point and is now gone. And you can also look at a succession of what happens to the areas after they die and distinguish between death that occurred recently and the skeleton is still present or visible beneath a fuzz of algal turfs, and what died further in the past or less recently, and they call that old mortality. And it was very obvious that factors such as the size of the coral and its location on a reef and its exposure to waves and sediments and even the depth at which it was growing were all confounding influences on what the life expectancy of any particular part of a colonial coral was during its time in the water. Now, by spring of 1997, Bob was fundraising madly, both because it was now the International Year of the Reef and also because he knew he was going somewhere with these surveys. And he had convinced Bob Stenek to firm up some methods for him to go and study these corals. And Bob had roped me in, and we had convinced him. He also had to look at the algae, the benthic algae that grow on the skeletons in greatest abundance, where herbivorous fish, and particularly the sea urchin Diadema, which by now had cratered and died throughout the Caribbean, and to add that to the corals that he was already focused on. And we were thinking of doing this on the 4 reefs at depths of about 8 to 12 meters in the zones of largest accumulation of big massive corals. So we're still thinking about big corals. And later that year, Phil Kramer, who by now was postdocing with Bob and responsible both for International Year of the Reef activities as well as getting what became the AGRRA program off the ground, his wife Patricia Kramer and Vladimir Kosmygin went out, field tested our methods at Carysfort, and then went over to Andros and carried out a large series of surveys looking at the benthos. And halfway through the Andros trip, they pulled in a reef fish volunteer named Ken Marks, who came and did a whole bunch of the fish surveys for the first time that we had previously proposed adding to the protocol. And they tweaked the methods as they went. And even before they left the Keys, they realized, well, they had to bring in patch reefs, and because that's where most of the live corals were now in the Keys, and also the reef crest environment where the palmata, before the acropores had all died, had been such a significant component of the reef fauna. So right from the very start, our surveys were stratified by habitat zone. that fall, we were happy enough with the protocols to put them out online as version 1. The following spring, Bob organized, with this money that he had been collecting, an international workshop which talked about them and tweaked the protocols. They became version 2. The Kramers and Vladimir went back to Andros along with Ken Marks and another fish biologist, and did a second large series of surveys that summer. And a couple of our colleagues from the first workshop went out and tried to do them as well. And I have to make a quick aside now to talk about the International Year of the Reef, which was going on simultaneously to this, because Bob had spent a lot of effort on that. And after it was over, he continued to reach out to the public with the teaching materials that he had developed during that period. So he'd already made a choral dance, and which he required us to, which he required us to perform in a Cunayala village when we were conducting surveys in Panama several years later. He was still making the attendees to a coral reef task force meeting in Florida perform his coral reef story play, and 8 years later, he was still showing his Cities Under the Sea slideshow that he had developed with Stephen Lutz during a training workshop for AGRRA in Barbados. So he never lost his commitment to that kind of outreach education, as many of you know. All right, well, by the spring of 1999, Bob was still fundraising, and that money was being used for training workshops and to give small grants to large numbers of small groups of people who went out and fanned around the Caribbean, the Gulf of Mexico, and even Brazil, and started collecting data using the survey protocols. The next year, there was another international meeting. The methods were tweaked again as a result of experiences gained regionally and came out as version 3. The following summer, there was the first of the large surveys, and this was 3 surveys in different parts of Cuba, which resulted in a total of 160 sites being assessed rapidly, and the following year in Panama, another 81 sites. So things were just, somebody said that Bob slowed down after the 70s, but from my perspective, he was still chugging along with great vigor in the 90s and early 2000s. In 2003, a large volume of the Atoll Research Bulletin was published summarizing the results of all these early studies that he had helped to finance. And Phil Kramer had a seminal review in there in which he presented what he called a reef health index based on a similarity diagram of a large number of the indicators that were collected by the surveyors for the sites that had been surveyed. And if you look at it, you'll see that there's a red star at the worst end of the diagram, and that was the Andros Reef that had been resurveyed in 1998. And what I forgot to say at the time is that that was just at the start of the big 98 bleaching mortality event. And Bob had been there in the Bahamas, and he saw the beginning of that event. And it surely was beginning to affect his thinking about what the status of coral reefs really is. By the spring of 2004, Rodrigo Garza was now postdocing with Bob, and he came out with another version, and which we used in the following year at the Dry Tortugas with Mark Palmer, who I don't know if he's still here, but thank you for allowing us to use the boat for that. And then in 2005, we taught that at a this version 4 at a workshop in Barbados. And that was the last time that Bob was actively engaged in one of the workshops. And you can see that he was an eager participant. And here he is at the end giving out one of our teaching certificates. And look at the grin on his face. He really enjoyed being in the field with us and stimulating people around the Caribbean, which is what this was allowing us to do, to get enthusiastic about taking responsibility for looking after the reefs in their areas. That fall was a huge mass bleaching event in the eastern, northeastern Caribbean. In response, Ken Marks and I went around focusing only on what was happening to the corals. This was a new protocol called AGRRA. Bob was with us. He saw firsthand the mortality that was resulting immediately after this huge immersion to unusually warm water. And I think that's probably when he really accepted the fact that humans were having a regionally significant effect on the status of coral reefs in the Caribbean. And by then he had also seen in so many of the areas where he had gone how depauperate the fish populations were. And had to notice the macroalgae and the other competitive organisms that were overgrowing the corals as they died from diseases or bleaching or competition. In 2007, Jerry Alt's group presented us with the results of a statistical survey they had done of our methods, proposed a lot of changes, which we agreed, and that made eventually version 5, which is the version we still use today. And just as importantly, because Bob was always interested in putting dots on a map, and how many dots did we have to put on the map to find out what it would take to assess the Caribbean. And Jerry Alt's analysis, using the Millennium maps that the TNC had made available at the time, was that, well, you'd need about 3,000 if you wanted to do a minimum of 2 habitats on the likely reef areas that are here. And I think we realized at that point we were never going to do it all by ourselves. In 2008, this was the last time that Bob went into the field with Ken and myself, and it was a short survey in New Providence, Rhode Island, and unfortunately we don't have any photos of it, so I'm very sorry, but I'm going to sort of back up now and more look at AGRRA as a whole and say, not that Bob wasn't, very much in our lives when we were in Miami, but he wasn't out in the field with us. We're still here. AGRRA is still collecting masses of data. As you can see, that was last year's total up through mid-December. Our methods are always online, and we now have a new website, thanks to some money that we got from Bob, and we're able to hire a professional for a change. Because our methods are online, where they can be easily adapted or adopted for use by any researchers or managers or conservationists or teachers. The reefs have changed in the last 20 years, and our understanding of how they're organized has changed in the last 20 years. And we have had to make some changes in the last 20 years. And even with the version 5 that we're now using, we still have to tweak it occasionally in order to accommodate the current changes. So I should stop for a minute and step back and say, well, what are the goals of the program? And right from the start, they have been to rapidly assess the key structural and functional attributes of tropical Western Atlantic coral reefs, and also provide fisheries-independent estimates of commercial fishing activity. We did not intend to address biodiversity. We left that to the people who were doing monitoring, because they take careful notes of everything that's on the bottom if they're looking at the benthos, or every fish that's swimming in the water column. But along the way, we have occasionally had the good fortune to add some primary information that wasn't known before. And this paper was published last fall by Ben Victor Benjamin and Ken Marks on a new species of hamlet that Ken and another fish surveyor found on the northeast coast of Haiti when we were doing a series of surveys in Fort Liberté Bay 3 years ago. We added lionfish to the fish surveys in 2007 when they turned up in Andros. And we have seen them, their emergence in our surveys throughout the Caribbean ever since. And that's, one example. If things change, you have to adapt. And that's all there is. Or you're irrelevant. It would be irresponsible not to collect information about lionfish nowadays just because we didn't know to put them in 20 years ago. Well, as you know, Bob's goal was to fill the data gaps for remote reef areas. And this map is almost up to date. We've only added a few more sites this year that were never seen before. The yellow dots represent surveys that were made before 2010, and the pink dots are the ones in the interval since then. We've had so much data coming in that Ken Marks has had to develop an online data entry tool, which is really neat because it picks up lots of mistakes that otherwise don't get detected with all its automatic QA/QC controls. And I've had to develop more complete habitat descriptions because we long ago discovered that there is a lot more to the Caribbean and the Bahamas than just reef crests, 4 reefs that are well developed at 8 to 12 meters with massive corals, and patch reefs. There are plenty of those, but there's lots of other kinds of environments as well. So that's kept us busy. What else has kept us busy over the last decade are developing training materials. A lot of them are online. That means that they can be used by anybody. We are told that a lot of them are just used in general reef ecology courses, all around the country. And some of them, like the quizzes, are restricted to when we are in the field actually conducting classes, training classes, which always have an in-water component as well as, a theoretical discussions in the classroom or the classroom equivalent. Sometimes it's a bar. And something that we have learned over the last decade is that with the more modern remote imagery, when it's good, you can use the modern statistical random sampling programs to really stratify your sampling surveys by habitat type. And when you do that, you discover that, well, not all reefs are created equal. And when sites are chosen representatively, sometimes they are naturally not very much hard grounds with small corals and lots of gorgonians or sponges, or they have been degraded as a result of our prior activities. And these photographs show the shoals in Samana Bay in the northern part of the Dominican Republic. We sampled at 11 randomly chosen sites 3 years ago, and the first 11 looked like this. They were mud banks with a few corals that were mostly being overgrown by very aggressive invertebrate animals, and they were, depressing. And our 12th and final survey site randomly took us to this. Huge colonies of very healthy looking, although slightly pale because it was a bleaching event, Orbicella colonies. And on the top of the shoal, which usually had nothing to speak of, was a big gorgonian field, and near that was that huge, was that huge stack of Acroporus cervicornis. Oh, I see, I missed a slide. Was that huge stack of Acroporus cervicornis. I think at the time it was the largest extant stand of staghorn coral anywhere in the Dominican Republic. And we found them totally as a result of this random sampling program that just took us there by fortuitous chance. So that's what we tell students when we train them, you know. When you're exploring, you don't know what you're going to find. And that's, of course, something that we got from Bob. And the other thing we have learned is that, you know, when random sampling doesn't work, it's okay to fill in your requisite number of sites with strategically selected locations, or sometimes you have to because the funding organization wants to know what is happening here off of some place of special interest. Well, we established AGRRA to do rapid assessments, and somewhere along the way, they somehow morphed in some places into monitoring, almost without my noticing it. And that was in part because we had found that you could repeatedly sample in the same site, even in this rapid fashion, and not collect a huge amount of data in any one site, but when averaged over the area of interest, you could record, you could see the record of, you know, momentous events. So this is 98 at the start of the mass bleaching mortality event on the reef crests in the Andros, and here it is on the forereefs to seawards of the reef crests, and you can see the plunge in the live coral cover. So it's now much lower than it was in the 90s, particularly on the reef crest, and not as much as in the deeper reef. But after looking at the new pattern for a few years, it was obvious that you didn't have to go out every year between catastrophes to have some idea of what would be there on the benthos. Now, if people are bringing in gill nets and suddenly fishing in an area that hasn't been fished before, that would also constitute a catastrophe. If you want to know what had happened, you better go back shortly thereafter. So for management purposes, over the years, with our colleagues, a large number of reports have been produced aimed at different audiences, but mostly policymakers or business people or conservation organizations. And I want to acknowledge Patricia Kramer because she has had a major part in many of these reports, and I show them here, and maybe there are others I don't even know about. And I want to remember Bob and say, because the one report in which she was the principal designer is particularly attractive artistically, and you know, Bob always appreciated good illustrations and beautiful art. So thank you, Patricia, for elevating our artistic and general appeal to the public. The first step in management and conservation is you need some biophysical information, and that's what the rapid assessments provide. Other organizations, like in this case, the Nature Conservancy, use the data from just 23 sites in 2013 to help build momentum in the Bahamas to get an area that is, at least in theory, protected of 2 million acres only a couple of years later. To effect real management, of course, takes a lot more effort with a lot of people, and you have to have all kinds of ancillary sociological and economic data, and you have to have buy-in from the fishermen, and you have to have compliance from anybody who is conducting any kind of development in the area, and you have to have controls on agricultural runoff, and that is a generational series of changes. But if you have enough people working together and collaborating and generating ideas about what to do as a group in a way that mimics how Bob would like us to do science in general, you can start to achieve wonders. And I think the best example for the Caribbean area is the Healthy Reef Initiative in Mesoamerica, the four countries of Mexico, Belize, Guatemala, and Honduras, which has been conducting AGRRA assessments every 2 years since 2006. have put out a series of reports, and Melanie McField, who started this, and Patricia Kramer, collectively spent several years working, adapting the report card concept of conveying scientific, complicated information in easy to understand visual colors for non-scientists. And the color scheme, it's like a traffic light. Red means it's terrible, green means it's, good to go. And they used 4 AGRRA indicators. 2 from the benthos are just the cover of live corals and the cover of fleshy macroalgae, because when things grow on dead corals, it's more likely to be that than anything else. And 2 aggregate measures of fish, one being the primary herbivores that when they're present can feed on algae, now that we don't have any diadema, unfortunately, to do that for us. And the most commercially significant, usually of reef fishes, which are the grouper and snapper, although you have to recognize that most of the many parts of the Caribbean, those are so long gone from the menu that really parrotfish are not only the primary herbivores, but small parrotfish are also the most important commercially available fish. And for each of these 4, I've learned how to do this. For each of these 4 indicators, they assign grades depending on their values from good to bad. And you can average these 4 grades and come up with a simple pie chart saying for each of the indicators, what is its grade and what is the overall grade for the reef site or the reef area or the country or the region as a whole. And they've now done this often enough that you can start to see regional changes over time. And they've been successful enough with their outreach to policymakers and communities that actually, some of these indicators are looking like they're beginning to improve, which is wonderful. And you can also parse them by country, and some countries are better in some indicators than others. So kudos for them for showing us the way. Okay, I'm going to skip to this one. I mentioned that that some of these improvements involve the fish, particularly the herbivorous fishes, and it's not fair to displace fishermen without providing them with some kind of alternate livelihood. You can't expect them to just go off and starve. And so what I'm also really encouraged about in Mesoamerica are some of the outreach efforts to try to provide other ways for fishermen to remain active on or near the water, which is all they really know to do and what they love. So this is one example from Utila in the Bay Islands, where tourists are being actively encouraged to engage in activities that will hire former fishermen. You can also extend that reef health indicator idea to more attributes that we actually collect with the AGRRA methods, and I'll just explain one very quickly for the benthos. If you just look at live coral cover, which is supposed to be the gold standard for what's on a reef, yellow in these different graphs, nowadays it's pretty miserable across the Caribbean. It's nothing close to what it was when I was cutting my teeth and learning how to scuba dive. But if you also add in the calcareous, the crustose coralline algae that help to bind reefs together, and some of them can provide substratum sites for coral larvae to settle. And if on top of that you add the area that's occupied by sparse turf algae, which are the first in quite often after coral tissues die, those ones tell you, well, there are herbivores around to keep them sparse because they're feeding on them, and also there's enough space between them for the coral larvae to have a chance of settling on the dead coral. So if you combine these 3 components of the benthos, you start to get a total which is looking a little bit more encouraging. You can say, well, 30 to 50% of the bottom is not doing so badly. There's potential for recovery. Unfortunately, nowadays, if you do the same thing with the organisms that we know compete successfully for corals, they add up to even more normally. But you can see that the light green at the bottom are the fleshy macroalgae, and they're the most important component regionally and subregionally. But there are other organisms out there which are in their own way also impacting the likelihood that corals will not survive if they happen to encounter them in the field. And you can take the ratio of the good things, the desirables and the undesirables, or the reef promoters, from the perspective of a coral. And the organisms that collectively conspire to, or collectively end up killing corals. And if it's less than, one, then you have reason to be a little bit concerned about the future of your reef. And now I want to finish up by just saying a few of our more recent outreach efforts, and these have been mostly driven by Patricia Kramer, working with the part-time GIS lady we have called Lynn Ross. And one of them is that they're in the monitoring partnership of the Coral Restoration Consortium. And there was, this is where the energy is in the coral field at the moment. Nurseries and outplanting sites are springing up spontaneously, almost it seems, all around the Caribbean. It's a train that has left the station and is attracting a lot of excitement and attention. And they are helping learn, well, where are these sites located? What are people doing? What's the purpose of the restoration? And you can see the answer to that is that mostly they haven't been able to find out. And so they're helping to organize the people who are, the group that is trying to collate and collaborate and organize the people who are engaged in these activities scattered around the Caribbean. And then there are lots of them. You can see on this GIS map they've produced that the pink dots represent the I can't read it now. The pink dots represent the nursery sites, and the black dots represent the sites where things have been outgrown. I think that's right, Patricia's nodding her head. And also on that map has been plotted, well, the benthic index for any place in the Caribbean for which we have calculated that ratio. So the green squares represent good reefs, and from the perspective of potential for corals to grow. And the red ones represent reefs that don't at the moment have much potential for corals to grow. And that's a good thing to know if you're starting to think about outplanting small corals to make a new reef. So it has a lot of practical value. They're also working on the database working group of the AGRRA Recovery Implementation Plan, which has been on the books since the AGRRAs were declared threatened about a decade ago. And one of the an example of one of the activities they're engaging in is that there's a large database emerging of the molecular, genotypic information about individual colonies. And there's a lot of information. There's a big inventory of where AGRRAs have been turning up around the Caribbean in the last 3 decades, because although they were thought to be down and out in the last few decades, we have found a few places where it looks like they never really disappeared. Or if they did, they are now starting to recover on their own. And these two sets of information shouldn't live